For more than a century, human origins were taught as a clean story, a straight line, one ancestral population, one moment of emergence, one successful migration out of Africa that replaced everyone else. It was elegant, it was easy to visualize, and it felt reassuring. Modern humans appeared, spread across the planet, and all other archaic humans quietly disappeared. That story shaped textbooks, documentaries, and entire generations of education. But today, genetic evidence is forcing scientists to confront a disturbing possibility. That story was wrong. Not partially wrong. Not missing a few footnotes. But fundamentally misleading. Because when scientists began to read the human genome carefully, not just as a record of ancestry, but as a fossil archive written in DNA, something unexpected emerged. Modern humans are not the result of a single origin. They are the result of contact, mixing, repeated encounters between different human populations. What we once imagined as a clean evolutionary tree now looks more like a tangled network. Branches that reconnect, lineages that merge, separate, and merge again. Populations that never fully replaced one another, but interacted. This changes everything. For decades, paleoanthropology searched for the ancestor, the missing link, the moment when one species became us. But genetics tells a different story. A story where there was never a single us, to begin with. Instead, there were many human groups, closely related, partially isolated, sometimes separated by geography, sometimes reunited by climate shifts, all contributing fragments to what we now call Homo sapiens. Human evolution, it turns out, was not linear. It was messy, and that messiness is written into every modern human genome on Earth. The illusion of a straight line, the idea of linear human evolution, is deeply intuitive. It mirrors how we like to understand progress, simple beginnings leading to complex outcomes, primitive forms giving way to advanced ones, old replaced by new. For much of the 20th century, fossil evidence seemed to support this view, or at least, it could be arranged to appear that way. A skull here, a jawbone there, a timeline connecting them. But fossils are rare, fragmented, and selective. DNA, by contrast, is everywhere, inside living people. When geneticists began comparing entire genomes across populations, they noticed patterns that could not be explained by a single origin. Some segments of DNA told one story, others told another, and these stories did not align neatly. Certain genetic markers appeared suddenly, not gradually. Others seemed older than expected. Some regions of the genome suggested deep African ancestry, while others hinted at contact with archaic populations long thought extinct. At first, these anomalies were dismissed as noise, statistical artifacts, contamination, misinterpretation. But as data accumulated, the anomalies refused to disappear. Instead, they multiplied. A forbidden idea returns. For a long time, the idea that modern humans interbred with other human species was considered speculative, even embarrassing. Early paleoanthropologists preferred replacement models, one superior population expanding and erasing all others. It fit cultural expectations of dominance and progress. But genetics does not care about narratives. It only records what happened, and what it recorded was unmistakable. Modern humans carry genetic material that did not originate within a single ancestral population. This means that when anatomically modern humans encountered other human groups, they did not simply compete or replace them. They interacted. They exchanged genes. They had children. Human history, at its biological core, is a history of encounters, not clean victories. The network model of humanity. Today, many geneticists describe human evolution not as a tree, but as a network. In this model, populations diverge, but they do not remain isolated forever. Climate changes open migration routes. Ice ages push groups together. Technological advances allow long-distance movement. When populations meet, genetic boundaries blur. Some encounters are brief. Others last generations. But even rare mixing events leave permanent signatures in DNA. Over tens of thousands of years, these interactions accumulate. The result is not a single ancestral line, but a mosaic. Each modern human genome is assembled from multiple ancient sources, layered across time. No population is genetically pure. No lineage is isolated. No origin is singular. This realization is deeply unsettling because it undermines a comforting assumption that there was a clear starting point, a moment when humanity truly began. Instead, humanity appears to have emerged gradually, through overlap and exchange. We did not simply replace other humans. In many cases, we absorbed them. Why this was so hard to accept? The resistance to this idea was not purely scientific. It was psychological. Linear evolution offers clarity. Networks introduce ambiguity. Trees allow us to rank. Networks force us to connect. Accepting a tangled origin challenges ideas of uniqueness and separation, not just biologically, but culturally. If humanity emerged through mixing, then difference is not a deviation from the norm. It is the norm. Variation is not an accident. It is the product of our deepest history, a question that changes the past. This leaves us with a question that scientists could not have asked a century ago. If modern humans emerged through repeated encounters with other human populations, then who, exactly, were those populations? Where did they live? How often did they meet us? And how much of them still lives inside us today? Because once we abandon the illusion of a single origin, the past does not become simpler. It becomes far richer and far stranger. And to understand it, we must follow the genetic traces backward into a world where humanity had many faces, many homes, and many paths forward. In the next part, we begin that investigation, not through myths or bones alone, but through the silent evidence carried inside our own cells. To understand how humanity truly emerged, scientists had to abandon the traditional tools of the past. Bones could only take them so far. Fossils reveal shape. They suggest movement. They hint at relationships. But they cannot tell us how populations interacted. They cannot reveal whether two groups merely coexisted, or whether they shared bloodlines. That limitation haunted paleoanthropology for decades. Then genetics changed everything. DNA as a historical archive. Every human cell carries a complete copy of our genome, more than 3 billion letters long. At first glance, it looks like chaos. But to geneticists, DNA is not random. It is layered. Older mutations sit beneath newer ones. Rare variants mark ancient events. Shared segments reveal shared history. When scientists compare genomes from different populations, they are not just measuring similarity. They are reconstructing timelines. They are asking, when did these groups diverge? When did they reconnect? And what survived those encounters? Unlike fossils, DNA records contact, not just ancestry. And once researchers learned how to read it, a pattern began to emerge. The shock hidden in the genome. The first major surprise came when scientists compared modern human DNA with genetic material extracted from ancient remains. They expected difference. What they found was overlap. Not complete overlap, but unmistakable fragments. Small segments of DNA in modern humans matched sequences that could not have originated within a single Homo sapiens population. These sequences pointed to other human groups, ones that had been physically distinct, geographically separated, and previously considered evolutionary dead ends. The implication was unavoidable. Modern humans carry genetic material inherited from other humans. Not metaphorically, literally. This was not a single anomaly. It appeared repeatedly, across populations, across continents. And once researchers knew what to look for, they found it everywhere. Interbreeding was not an exception. For a long time, interbreeding was treated as an accident, a rare event, an evolutionary footnote. But genomic data revealed something else. The genetic signatures of ancient admixture were too widespread, too consistent, to be dismissed as rare mistakes. They followed clear geographic patterns. Populations whose ancestors left Africa earlier carried different archaic segments than those whose ancestors remained longer. Groups in different regions showed distinct layers of genetic inheritance, as if each migration wave picked up traces from the populations it encountered along the way. This suggested something profound. Interbreeding was not an anomaly. It was a recurring feature of human expansion. Whenever populations met and remained in contact long enough, genes flowed. Competition did not exclude intimacy. Cultural difference did not prevent reproduction. Biological boundaries were porous. Human groups did not behave like isolated species. They behaved like neighboring populations. Why mixing left a permanent mark? From an evolutionary perspective, most interbreeding events disappear. Genes dilute. Variants vanish. But some genetic material persists, especially when it provides an advantage. Geneticists discovered that certain archaic DNA segments were overrepresented in modern populations. They survived selection. Why? Because they helped. Some variants influenced immune responses, improving resistance to unfamiliar pathogens. Others affected metabolism, altitude adaptation, or skin physiology. These genes were not random leftovers. They were useful. This means that contact between human groups was not merely neutral. In some cases, it shaped survival itself. Modern humans did not just absorb other populations biologically. They integrated solutions evolved elsewhere. Humanity, at its core, is adaptive because it is composite, a new way to think about human expansion. This forced scientists to reconsider one of the most familiar narratives in human history, migration. For generations, migration was imagined as replacement. One population moves in, another disappears. Genetics tells a subtler story. Migration often meant incorporation. Incoming groups absorbed local populations. Local populations absorbed newcomers. Sometimes one group dominated culturally, while the other contributed genetically. Sometimes the reverse happened. Over time, identities changed, but DNA preserved the record. The genome became a palimpsest, new stories written over old ones, never fully erased. The limits of identity. This discovery also raised an uncomfortable question. If modern humans are genetically mixed, then what does it mean to define a population? Genetic clusters exist, but they overlap. Lineages persist, but they intersect. No group represents a pure continuation of a single ancestral population. Instead, each population is a statistical center, surrounded by gradients. Boundaries are fuzzy. Transitions are gradual. Human diversity is not the result of separation alone. It is the result of repeated contact, from evidence to implication. By the time this genetic picture became clear, the old linear model could no longer survive. Human evolution was not a ladder. It was not even a tree. It was a braided stream. Paths diverged and rejoined. Populations separated and merged again. Extinction was not always disappearance. Sometimes it was absorption. This realization did more than rewrite textbooks. It changed the very definition of what it means to be human. If our species emerged through interaction, then humanity is not defined by isolation or purity. It is defined by connection. What comes next? But genetics does not stop at identifying mixture. It can measure how much. It can estimate when. And it can reveal who contributed what. As scientists refined their tools, they began to distinguish different layers of ancient ancestry within modern genomes. Some layers were shared widely. Others were region-specific. And these layers point to particular human groups, each with its own story. To understand how deep this complexity goes, we must examine those groups individually. Who were they? Where did they live? And how much of them still lives inside us? That investigation takes us deeper into the human past, into encounters that wresh amendments our species forever. Once geneticists accepted that modern humans are a composite species, a new question became unavoidable. Composite of whom? The genome did not just reveal that interbreeding happened. It preserved clues about with which populations, and under what conditions. These were not abstract ancestors. They were humans who lived, hunted, adapted, and survived in environments modern humans were only beginning to enter. And in several crucial moments, they left behind more than fossils. They left behind themselves. Meeting other humans. When anatomically modern humans began expanding beyond Africa, the world was not empty. Eurasia was already inhabited. Different human populations had lived there for hundreds of thousands of years, adapting to cold climates, seasonal scarcity, and unfamiliar ecosystems. These groups were not evolutionary failures. They were specialists. They knew the land. And when modern humans arrived, they did not encounter an evolutionary void. They encountered neighbors. Genetic evidence shows that these meetings were neither singular nor brief. They occurred repeatedly, across thousands of years, in different regions. Sometimes contact was limited. Sometimes it was sustained. But in every case where contact lasted long enough, genetic exchange followed. What the genome reveals. Modern human DNA contains segments that stand out clearly against the background of African origin ancestry. These segments are older, more divergent, and clustered in ways that only make sense if they came from populations that had been separate for long periods of time. Crucially, these segments are not evenly distributed across the genome. They concentrate in regions linked to survival, genes related to immune response, skin physiology, metabolism, environmental tolerance. This pattern is not random. It suggests selection. When modern humans entered new environments, they encountered unfamiliar pathogens, climates, and dietary pressures. Local human populations had already adapted to those conditions. Interbreeding allowed modern humans to acquire those adaptations rapidly, without waiting for slow mutation and selection. In evolutionary terms, this was an extraordinary shortcut. Humanity did not just spread geographically. It borrowed locally evolved solutions. Absorption, not erasure. For a long time, extinction was assumed to mean disappearance. But genetics forces a different interpretation. Some human populations did not vanish completely. They were absorbed. Their cultural traditions may have faded. Their languages disappeared. Their physical forms changed. But parts of them survived, quietly, inside the genomes of those who came after. This reframes extinction itself. In human evolution, extinction was sometimes biological death, but sometimes it was transformation. A population could cease to exist as a distinct group, yet continue as a genetic contribution. Modern humans are living archives of these absorbed populations. Why not everyone inherited the same past? One of the most striking findings in genetic research is how uneven this inheritance is. Different modern populations carry different mixtures of ancient DNA. This reflects geography. Groups whose ancestors traveled certain routes encountered certain populations, and not others. Populations that remained in Africa retained deep African diversity, shaped by long-term population structure rather than interbreeding with Eurasian groups. Populations that moved through the Middle East encountered different human groups than those who migrated north into colder regions. Later migrations layered new encounters on top of old ones. As a result, no two populations share the exact same genetic history. Humanity is unified, but not uniform. The cost of mixing. Interbreeding was not always beneficial. While some inherited genes improved survival, others carried risks. Genetic incompatibilities can arise when populations diverge for long periods. Some archaic variants were later removed by natural selection because they reduced fertility or increased disease susceptibility. The genome reflects this filtering process. Useful genes stayed. Harmful ones faded. This shows that interbreeding was neither purely positive nor purely negative. It was complex. Like all human interaction, it involved trade-offs. Rethinking modern humans. Perhaps the most unsettling implication of this research is what it says about the concept of modernity. Anatomically modern humans are often treated as a sharp biological category, a clean break from what came before. Genetics does not support this. Instead, it suggests continuity. Modern humans emerge gradually, through interaction, not replacement. There was no single moment when humanity suddenly became modern. There was overlap, exchange, blending. Traits we associate with modern humans, resilience, adaptability, geographic range, may themselves be the product of mixture. Modern humanity did not arise despite interbreeding. It arose because of it. The deep irony of human origins. For centuries, human societies emphasized purity, of lineage, culture, or identity. Yet biologically, humanity is the opposite of pure. Our species exists because boundaries were crossed, because encounters occurred, because difference did not always lead to separation. The genome tells a story of openness, sometimes forced, sometimes accidental, sometimes cooperative, but always consequential, from global pattern to specific histories. By this point, the broad outline is clear. Human evolution was shaped by repeated contact between populations. But this global pattern hides important local stories. Some populations experienced extreme isolation after mixing. Others expanded rapidly from small founding groups. These processes left distinctive genetic signatures, amplifying certain traits, increasing others, and sometimes creating vulnerability. To understand how mixture and isolation interact, scientists turn to specific case studies. Among the most revealing is one involving a population shaped by migration, bottlenecks, and intense genetic inheritance. A case where history, culture, and DNA align in unexpected ways. That case is explored next. Interbreeding explains how humanity became mixed, but it does not explain why some populations look genetically unusual. To understand that, scientists had to examine what happens after contact. When movement stops, populations shrink, and history applies pressure not through expansion, but through constraint. This is where isolation enters the story, and with it, one of the most powerful forces in population genetics, the founder effect. What happens when a population shrinks? Imagine a large population with immense genetic diversity. Now imagine that population reduced to a few hundred, or even a few dozen individuals. War, climate collapse, migration barriers, persecution. History repeatedly creates these moments. When only a small subset survives or relocates, they carry with them only a fraction of the original genetic variation. Whatever genes they happen to carry become amplified in future generations. This process is not intentional. It is statistical, and it reshapes populations faster than natural selection ever could. This is known as a genetic bottleneck. The founder effect explained. When a new population forms from a small number of individuals, those individuals become founders. Their genetic traits, common or rare, define the future population. If a rare variant is present in one founder, it can become widespread simply because there are no alternatives. Over generations, this creates genetic signatures that appear unusual, exaggerated, or highly concentrated. The founder effect does not require isolation forever, only long enough for a small gene pool to expand internally. Human history provides many such cases, but few are as well documented or as instructive as populations shaped by repeated migration and enforced separation. Isolation after mixing. This is where the story becomes especially important. Interbreeding increases diversity. Isolation reduces it. When these two forces occur sequentially, they produce striking results. A population may carry genetic material from multiple ancient sources, but express it in a narrowed, intensified form. Certain lineages dominate. Others vanish entirely. Over time, this creates genetic coherence, not because the population was ever pure, but because alternatives were lost. From the outside, such a population may appear homogeneous. Internally, it carries a deep, layered history. Genetic diseases as historical markers. One of the clearest indicators of founder effects is the prevalence of certain inherited diseases. When a small population expands in isolation, harmful recessive mutations can become unusually common. These conditions are not the result of moral failing or biological inferiority. They are historical artifacts, markers of past bottlenecks. Geneticists began to recognize that studying these diseases could reveal something else. Population history. By tracing the distribution of specific mutations, researchers could reconstruct migration paths, estimate population sizes, and identify periods of extreme isolation. In this way, illness became evidence, not just of biology, but of history written in DNA. The paradox of vulnerability and insight. There is a deep irony here. The same isolation that increases genetic risk also makes populations invaluable for research. Because their genetic variation is constrained, patterns are easier to detect. Signals are clearer. This has allowed scientists to identify genes associated with neurological disorders, metabolic conditions, and immune responses more quickly than in genetically diverse populations. In other words, historical suffering inadvertently accelerated modern medical understanding. This does not justify the suffering, but it does show how deeply intertwined biology and history truly are. Culture as a mirror of genetics. Interestingly, Genetic patterns shaped by founder effects often mirror cultural patterns. Languages simplify and stabilize. Rituals become highly conserved. Identity becomes tightly defined. This is not coincidence. Isolation affects culture and genes simultaneously. Both are transmitted within the same limited population. Over generations, this produces a powerful sense of continuity, even as the deeper genetic history remains complex. From the outside, such communities may appear ancient and unchanging. From the inside, they are the result of adaptation under pressure. Why this matters for human origins? Founder effects remind us that human genetic patterns cannot be interpreted without history. A genetic signature may look ancient, but actually reflect recent isolation. Another may look unusual, but simply be amplified by chance. Without context, DNA can mislead. With context, it becomes extraordinarily precise. Understanding this distinction is essential when comparing populations across regions and time, because not all differences reflect deep ancestry. Some reflect survival, beyond the main narrative. By the time geneticists fully understood the power of bottlenecks and founder effects, they realized something crucial. Not all human populations fit neatly into the major migration narratives. Some communities emerged far from expected regions. Others preserved identities despite minimal genetic continuity. These exceptions challenge simple equations between DNA and identity. They force a deeper question. What defines a people, genes, culture, memory, or choice? To answer that, scientists had to look beyond the main corridors of migration to communities that defied expectation. That journey takes us to the edges of the map and to populations whose stories reveal the limits of genetic explanation. By the time the genetic evidence is fully assembled, one conclusion becomes impossible to avoid. Human origins are not a single story. They are a layered process. A long sequence of encounters, separations, reconnections, and survivals unfolding across tens of thousands of years. What once looked like a clean beginning now reveals itself as a slow emergence. Not a moment, a process. The end of the single origin myth. The idea that humanity began in one place, moved outward in a straight line, and replaced everyone else is no longer defensible. Genetics does not support it. Instead, it reveals a world populated by multiple human groups, overlapping in time, space, and biology. Some diverged early. Some remained connected. Some disappeared as cultures, but survived genetically. Modern humans did not arrive fully formed. They were shaped along the way, by climate, by geography, by contact, and by each other. This does not diminish humanity. It deepens it. Identity beyond bloodlines. One of the most important implications of this research is what it says about identity. Genes matter. They record ancestry. They trace movement. They preserve biological memory. But genes are not the whole story. Human groups have repeatedly shown that identity can persist even when genetic continuity weakens, and that genetics can remain even when identity disappears. Culture, language, and shared memory do not always align perfectly with DNA. Sometimes they reinforce one another. Sometimes they diverge. This does not mean one is false and the other true. It means human identity operates on multiple layers simultaneously. Biological, cultural, historical. Reducing identity to a single layer, whether genetics or tradition, oversimplifies what humans actually are, what the genome cannot tell us. Despite its power, DNA has limits. It can show that populations mixed. It can estimate when. It can suggest where. But it cannot tell us how people understood those encounters. It cannot reveal whether mixing was peaceful or forced, whether it was rare or routine, whether it was welcomed or resisted. Those stories belong to archaeology, anthropology, and history. The genome is silent on meaning. It only preserves outcome. That silence is important. It reminds us that science explains mechanisms, not experiences. A species defined by contact. Still, when viewed as a whole, the genetic record delivers a clear message. Humanity is not defined by isolation. It is defined by interaction. Our species expanded not by remaining separate, but by crossing boundaries. Geographic boundaries, biological boundaries, social boundaries. Even when those crossings were brief, they left lasting marks. Every modern human carries fragments of this shared past, not as symbols, as biology. Why this story matters now. Understanding human origins is not just an academic exercise. It shapes how we think about difference today. If humanity emerged through mixture, then diversity is not a modern problem to be solved. It is an ancient condition to be understood. If no population is biologically isolated, then claims of absolute separation collapse. And if identity has always been layered, then simple answers about who we are will always fail. This perspective does not erase difference. It contextualizes it, from the past to the present. In many ways, the human genome acts like a quiet archive. It does not argue. It does not persuade. It simply records. Across billions of letters, it preserves the outcomes of choices, movements, and encounters made by people who never imagined they were shaping the future of an entire species. Their lives were local. Their impact was global. Modern humans are their cumulative result. A final reflection. For centuries, humans searched for a single origin, a moment that explained everything. What genetics offers instead is something more honest. A history without a clean beginning, without clear boundaries, without purity, but also without isolation. The story of human origins is not a story of replacement. It is a story of persistence through connection, and it is still unfolding. Because the same forces that shaped us in the past, movement, contact, adaptation, continue to shape us today. The difference is that now, for the first time, we can see their traces clearly. Not in myths, not only in bones, but written quietly inside ourselves.